All right, we're gonna get underway with our fourth installment of the Summer Genetics Lectures. And today we're gonna pick right up where we left off. So we have now discussed our three major players in genetics. We discussed DNA as the genetic, the molecule that's gonna store the genetic information, which is the recipe for how to make all the proteins in the cell. And we discussed the mechanisms by which DNA can replicate itself. This is that ever elusive self-replicating molecule that people were looking for after Darwin's publication. We also should now be familiar with how RNA is formed in the process of transcription using the DNA as a template. And here we discussed how we're going to take the information, the recipes stored in the DNA for how to make a particular protein, and that gene is going to be transcribed into RNA, which has an RNA copy of that recipe, which is then taken to the ribosome, where that recipe will be translated into the protein that was encoded. And we discussed how some of the accessory RNAs like tRNA and rRNA, as well as things like microRNAs, can help regulate this process. So that is the big bulk of how everything in genetics or in life in general is going to work is through this. I said this is kind of the secret to life right here. So almost everything a cell does somehow is related to this process, and any differences between different organisms or different cells within an organism comes down to the proteins that are produced in that cell. So the first, now that we have those basic concepts laid out for us, the first major thing that should come to light then is we discussed that the process of transcription, the gene knows where to start and stop the transcription of the gene due to some sequences that are built right into the DNA. So in this case, we discussed that there's a promoter region that is within the DNA, and that's going to tell the cell where to begin transcription. And then we have a start codon within the mRNA that'll tell the ribosome where to start translation. So if those are built into the DNA, these sequences are directly built in to tell it where to begin transcription and translation, then we have our first major thought exercise here. Because if you look here on this figure on the left over here, this cell right here, this is a human neuron, and that is a single cell, right? It is one single cell that is relatively large, has these complex branching networks to it. Below that, this little guy right here, this little red guy, that too is a single cell from a human. This is a lymphocyte. This is a blood cell that is involved in your immune response. So both of these are human cells. They're cells found in a human. And these are both single cells. And you can see morphologically, these cells are entirely different from one another. I mean, both on the size scale as well as the morphological structure scale, these are entirely different cells. And if we said that what gives a cell or an organism its qualities, its phenotype, is the proteins, then we would have to surmise that these two cells, since they look so different, they have different qualities, they must express different proteins. And we would be correct in assuming that. In fact, if you look at some different cells found within a human, here's a neuron. My drawing does not do justice to what the actual neuron looks like, but there's a neuron. Neurons, it turns out, produce a protein from their DNA through the processes we described in the previous lectures, called cholinesyl transferase, or CHAT. And as it turns out, neurons are the only cells in the human body that produce that protein. Right next to that is the beta cell found in the pancreas. And the beta cells of the pancreas are the only cells in the entire body that produce a protein called insulin, which is a hormone. We discussed insulin a few times in our previous lectures. And just for comparison, here's a smooth muscle cell from a human, and it turns out that smooth muscle cells are among the only cells in the body that produce a protein called calponin. So we can see then our idea is correct. These are 3 different cells found within a human, all of which are producing different proteins. So if we back it up then, we can say that the transcription of a gene by RNA polymerase is going to be mediated by the promoter region, which is part of the DNA. So you can see here, this gene has a promoter region, in this case contains a TATA box, and your general transcription factors, things like TF2D, are going to come in here and recognize that TATA box and wind up recruiting RNA polymerase II, where they can then produce an mRNA. That mRNA that they produced has built into the gene itself what's going to wind up being a start codon and a stop codon, and those are going to be used to instruct the ribosome where to begin translating. So here we can see here's our Kozak sequence on the RNA we just produced. It has a start codon right here that's going to bind to the tRNA carrying methionine, and that is going to establish our reading frame, and that is going to signal to the cell where to start translation. So if we have cells in the human body that are producing different proteins, but the signals that instruct transcription and translation are built into the DNA, then our logical conclusion would be then that all of the cells in the human's body must have different DNA, right? A beta cell must have different DNA than a neuron, which must have different DNA than a calponin, because that would be the only way that they can express different proteins if the signals that initiate transcription are built into the DNA. And that was kind of the mode of thought for how this must work once all of this was surmised. But science getting in the way here, it turns out that's not true. One of the seminal experiments that was done is you can take an adult frog, in this case, Xenopus, and scratch off a few stem cells from its back, just get some skin cells, and grow those skin cells in a culture dish. You can grow them in an incubator. And then from those skin cells, you can remove the nucleus. Remember, all of the DNA is stored within the nucleus of the cell. And then from a different frog, go ahead and remove an unfertilized egg from a female frog and remove the nucleus from that egg. So that egg no longer has any DNA. And then take the nucleus from the skin cell and transplant it into the enucleated egg. So now you have an egg from a frog whose DNA came from the skin of another frog. You grow this in an incubator, and voila, the next day, you got yourself a tadpole. So what this experiment, this is called somatic cell nuclear transfer, and what this tells us is that the DNA in any given cell of an adult organism provides all of the instructions needed to produce any type of cell in the entire organism, right? This was a skin cell from an adult frog, and that formed all of the different types of cells in the tadpole. This includes the pancreas, the neurons, the smooth muscle cells, and the skin cells. So the DNA within a skin cell knows, it has the instructions for making every single type of protein in the organism. And you can do this with anything. If you're going to remove a liver cell from the frog or whatever, this will all work the same way. All of the cells in the adult frog have the exact same DNA. So all cells in an organism have the same DNA. If you take my embryology class, we'll talk more in detail about why that is, but this is just a truth. If you are a vertebrate organism, then all of the cells have the same DNA. So that brings us then back to our, well, how in the heck, if everything has the same DNA, then that means every single cell, whether it's a skin cell or whether it's a neuron or a beta cell, has the TATA box sitting up front of the gene. So you're going to make insulin in every single cell in the organism. So the TATA box is going to be the same in every single cell in the organism. And then once you make that RNA, each of those RNAs are going to have the exact same Kozak sequence. So you're going to translate that insulin protein in every single cell in the organism. And we're right back to the how in the heck is this possible? Well, the answer, if you're sitting there anxiously waiting, oh my God, how is this possible, is that we can regulate gene expression at different levels. In fact, every level of the game that we've talked about so far is a level potentially by where the cell can regulate gene expression. So the pre-mRNA is transcribed from the DNA at the promoter. And it turns out we can regulate whether or not that happens or not. That pre-mRNA then has to be processed, right? We need a 5' 7-methylguanosine cap on it. We need a 3' polyadenylated tail. We need to splice out those introns, splice together the exons. We can regulate whether that happens or not. Once we wind up with our mature mRNA, it then has to be exported out of the nucleus, go to the cytoplasm. We can regulate whether that's going to work or not. And then once it's in the cytoplasm, the mRNA has a half-life. Remember, it can be destroyed or kept around. We talked about how those HNRNPs can bind to insulin, either prolong its half-life in the case of high blood sugar or high blood glucose levels, or it can decrease its half-life in the instance of low blood glucose. We can regulate that too. So we can regulate the stability of the mRNA, how long it sticks around in the cytoplasm. And then once that mRNA gets out into the cytoplasm, it has to find a ribosome where the ribosome can translate it. We can regulate whether that's going to happen or not. In fact, we already talked about one example of that with our microRNAs. Remember, your microRNAs bind to the mRNA and prevent translation. So we can regulate it there. And then remember, finally, after we translate the protein, it still has to be processed post-translationally. So it has to be folded into the proper orientation. And it also may have to be modified through proteolysis or phosphorylation, ubiquitination, acetylation, whatever. We can regulate whether that happens or not. So at each of the 6 different major steps we've talked about in the first 3 lectures, we can regulate whether or not these things happen to allow a gene to get expressed or not. Now, as it turns out, just because cells are really good at conserving energy, they don't want to waste energy. If you don't need a protein expressed in a cell, if you are, let's say, a neuron that's never going to need to express insulin, then the place we generally are going to regulate this process is up here at the first transcriptional level. So overwhelmingly, we find that transcriptional regulation of a gene is the overwhelming way that cells can do it. Although that's not to say all of these other mechanisms are used, but especially if you're not ever going to need a protein in a particular cell type, we can stop it at the transcriptional level. One, this conserves energy, right? If you sat through the first several lectures, you see how much crap has to go in to making a functional protein. So why waste all the energy transcribing and processing and transporting and translating if you don't need the protein anyway? It's a lot of energy you're wasting for no good reason. The other thing is, if you are a cell that is not supposed to express a certain protein, then expressing that protein is probably going to be detrimental to that cell. So if you want to make sure you're not going to screw up, it's better to stop this. It's better to regulate this early on. That way, if you accidentally transcribe a gene you're not supposed to make, you have all of these other steps that you can try and fix your problem and get rid of it. If you're waiting up here to step 5 or step 6 to turn this off and you accidentally screw up and make it, you have less chance to correct your mistake later. So we'll find that transcriptional regulation is overwhelmingly the go-to for a cell to regulate proteins that it doesn't need or doesn't want to make. So you're then right back at square one. But Dr. ZeRuth, if we just said that the promoter region is in the DNA and all cells have the same DNA, then how are we going to regulate this? If the ta-ta box is there in one cell, it's there in all cells. And that's true. And what it is I didn't necessarily lie to you, but I omitted some information in the earlier lecture on transcription when I said the promoter is responsible for turning a gene on. And that is entirely true. The promoter region that contains things like the ta-ta box and the initiator sequence is where your general transcription factors, TF2D and TF2B and TF2E, H, and F are all going to come in there to recruit RNA polymerase II. But there's more to the story than that, because there are other regions that surround the gene that help regulate whether TF2D can ever come into the core promoter itself. So that region we discussed in the transcription lecture that I called the promoter, that is more specifically called the core promoter. And then immediately upstream, remember upstream is away from the gene, away from the transcriptional start site, immediately upstream of the core promoter, you have this region here called the distal and proximal promoter. And these are, it's fuzzy as to where you draw the line to where these things start and end. In general, we think of the proximal promoter being immediately upstream of the core promoter, generally within the first, and again, this is gray area, but somewhere between 250 to 1,000 base pairs is usually deemed the proximal promoter. And then immediately upstream of that is the distal promoter. And this can be anywhere from 250 up to 2,000-3,000 base pairs upstream. It depends on the gene. There's a lot of gray area. So usually these get kind of grouped together by most of us, and we'll just call it the proximal promoter, or we'll call the whole thing the promoter. So I'm not going to expect you to draw a line as to where the proximal distal promoter are. They're somewhere upstream of the core promoter within some distance. So I usually kind of cut off that the distal promoter usually cuts off at about 1,000 base pairs upstream of the core promoter, but whatever, somewhere in that region. But they can be distinguished from the other element that is going to regulate the core promoter, and these are called the enhancers. And the enhancers, notice how the core, the proximal and distal promoter are kind of immediately abutting that core promoter here. So this whole thing is our general promoter region. The enhancers can be located literally anywhere. You can have an enhancer that is 300,000 base pairs upstream of the gene. You can have an enhancer that's 300,000 base pairs downstream of the gene. In fact, you can and often do have enhancers located within the introns within the actual gene. So introns often have enhancers located in them. And that enhancer within an intron can either regulate this gene that we're talking about, or it can regulate some other gene. So enhancers really can be located anywhere and are often great distances away from the gene that they actually regulate. So now we have this more complicated picture of a DNA regulatory element. You have your core promoter, which binds your general transcription factors, and then you have your proximal distal promoter and your enhancers, and these bind transcription factors. We're losing that word general there. A transcription factor is any protein that can bind to DNA motifs and regulate the activity of a gene. So these are not the same as your general transcription factors, whose job it is to come in there and recruit RNA pol 2. These are the supporting players that help regulate that process, the process of initiation we discussed at the transcription level. And it becomes a gray area drawing the line, but generally your general transcription factors are going to be termed things like TF2D, TF2E, TF2H. Transcription factors do not get those designations. They have whatever name the people that discovered them gave it. One of the transcription factors my lab is interested in is called GLIS3. So they're going to have their own unique names. So the way these work is they are going to bind motifs. Again, a motif within the DNA is just a sequence of bases. So, G, 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 C, G could be a motif that the transcription factor binds. And these are almost always going to be located in either the major or minor groove of the DNA. And we can classify transcription factors as either an activator, if it activates a gene or turns a gene on, or a repressor, if it represses a gene or turns a gene off. And there's two other ways to classify these then, and this is true for both activators and repressors, is they can be broken down as either a cis-acting control element or a trans-acting control element. And the major difference here is a cis-acting control element is some transcription factor, let's say here, that binds at the proximal promoter and activates a gene. So since it's binding, cis is the Greek word for same. So since it's Latin, so since it's binding to the same area as the gene, that's a cis-acting element. Now, the transcription factor itself is a protein. So it may be produced somewhere else on a completely different chromosome within the organism. So here we have a gene that gets transcribed into an mRNA and then translated into a protein. And then that protein goes and binds the cis element to control this gene. So this is my transacting element because I had to transcribe this RNA in order to make this protein, which then activates this gene. So it acts on a different, trans means different, it acts on a different region of the genome versus the cis acts on the same. So transcriptional activators will bind to the DNA, usually within the cis region of the gene it's activating, and they are going to, once bound to the DNA, have other regions that can then turn the gene on or help recruit RNA polymerase II to the core promoter. Transcriptional repressors will also bind to the DNA, but they will have mechanisms that prevent the recruitment of RNA polymerase II to the promoter region. So that means for a protein to have the job of being a transcription factor, you know, it's like you want to be a doctor, you got to have some qualities to you in order to be a doctor. You got to get your good grades, you got to, you know, do your shadowing, you have to be competitive in order to be a doctor. Not just anyone can be a doctor. Same thing with a protein here. So if you want to be a transcription factor when you grow up, you've got to have two qualities. Since transcription factors bind to DNA, and then once bound to DNA can either activate or repress transcription, they must have two different functional domains that allow this to happen. So a domain is a region of a protein that when cut away from the rest of the protein still does some job. Okay, so if you have a protein, if you have a particular region within that protein, and I can cut away the rest of the protein and just take that region in isolation will still do the same thing as the whole protein. That is a domain. And for you to be a transcription factor, you've got to have two different domains that allow you to do this. The first of these, obviously transcription factors come in and bind DNA and then either activate or repress transcription. The first domain you have has got to be a DNA binding domain. You must have a domain that allows you to bind to DNA. And the way this generally works is you're going to have, remember, proteins have those secondary structures like alpha helices and beta sheets and beta barrels. Well, generally the alpha helices of the protein within the DNA binding domain are going to interact with the major groove or sometimes the minor groove of the DNA, and this is going to allow them to interact with those bases. So if you will allow me to show you, remember I talked about in the DNA lecture, our very first lecture, that you have the DNA double helix is going to form what we call those major grooves and minor grooves of the DNA. So if this is my DNA, remember I have this sugar phosphate backbone that is over here that is basically blocking the bases that are held together in the middle of the DNA molecule. So if I'm a transcription factor with a DNA binding domain, my DNA binding domain is going to chemically interact with those nitrogenous bases, my A's, T's, G's, and C's of the DNA. So over here, I really can't get in there because the sugar phosphate backbone is kind of blocking those bases. So most transcription factors are going to bind right here at what we call the major groove of the DNA. And that's because here, where I have that major groove formed, I have direct access to these bases and I can bind to them by chemically interacting with them. So these are usually where we're going to see these transcription factors bind, is at that major groove for that reason. This is where they have access to the bases that are in the DNA. So that being said then, our transcription factors are going to have these DNA binding domains. Their alpha helices can get into those major grooves and interact with the bases of the DNA. There are a number of different DNA binding domains in eukaryotes that have evolved over time. They all do the same thing. They're all going to kind of interact with the major groove of DNA to interact with those bases, to stick to the DNA, chemically bind to it through generally weak hydrogen bonding. But how they go about that varies by the type of DNA domain there is. The most common DNA binding domain in the metazoans, the multicellular animals, is called the zinc finger domain. So these are going to have, they're going to form a DNA as the regions of the protein are going to interact around a centrally located zinc ion, a metal. This is one of the reasons you have to get zinc in your diet, is for these things to work. And this allows you to form, once the protein kind of forms around the zinc ion, they form little fingers that start sticking up. And those fingers are going to interact with the DNA. And each finger can represent, can recognize a particular DNA sequence. So one finger can recognize GG, and this finger can recognize CG, and this finger can recognize CC. So then all together it recognizes a sequence that goes, GG, CG, CC. So these are very modular in nature. There's a couple of different classes of zinc finger domains, but it's more detailed than you really need to know for the sake of this class. But for instance, there's what's called the C2H2 zinc finger domain. And what that means is each zinc ion is held together around the protein by 2 cysteines and 2 histidines. And these are the amino acids that are going to make direct contact with that zinc ion. In contrast, a C4 zinc finger domain has 4 cysteines that are making contact with the zinc ion, but generally achieve the same thing. So to give you an example of one of these looks like, this is the zinc finger domain from one of the proteins my lab is interested in. And what you're looking at here, this is the single letter codes for the amino acids that make up the domain. And again, this is a domain. So if I cut this region out from the rest of the protein, it'll still bind DNA. So here we'll start, this is the amino terminus of this protein, and we'll just kind of follow it around. So this protein, here's my first cysteine, and that's going to interact with the zinc ion. So the blue ball there is a zinc ion. And then it loops around to form one of these fingers. So that's the finger I'm talking about. And then here's my histidine that's going to interact with the zinc ion. And then that's going to have another histidine here that interacts with the zinc ion. And then you get this, what's called the interdigital region coming up here. And then it's going to, another cysteine is going to interact with the next zinc ion, loops up here. This cysteine interacts with the zinc ion, forms another finger. There's a histidine, forms with the zinc ion. There's another histidine, zinc ion, another interdigital region. There's my other cysteine. We can follow this around like this. And that's the carboxyl end. So this is one continuous protein, and each of these cysteines and histidines are going to interact with a different zinc ion. So these are the different modules I was talking about. So this is a C2H2 zinc finger protein. See, I got 2 cysteines and 2 histidines making contact with each zinc ion. So this is a C2H2, and it has 5 modular units. So there's 5 different fingers within this that are going to interact with the DNA. So each of these fingers is capable of interacting with the DNA. Remember, DNA, deoxyribonucleic acid, is generally negatively charged due to that sugar phosphate backbone. So what we usually see with the zinc finger domains then is the fingers that interact with the DNA are going to be magnetically drawn to the DNA because they have positively charged amino acids that can magnetically be attracted to the negatively charged DNA backbone. So if you notice my zinc fingers here, I have a positively charged lysine, a positively charged arginine, arginine, lysine, arginine. Those all have positive charges on them, so they're going to want to stick to the DNA. Look at my next finger. Lysine, 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 arginine. It's going to want to be drawn to that DNA. Here's a lysine, here's an arginine. Over here, I have a lysine, arginine, arginine, lysine. So those positive charges in those amino acids are going to want to stick to the DNA. And all together, then, you get something that happens like this. So I have my DNA double helix. Here's the major groove. And notice the alpha helix of the zinc finger domain is going to have contact with these bases, where it can form hydrogen bonds with the bases. And each one has some modular sequence. So here, one zinc finger has arginines that interact with a GG motif. Here I have my arginine and histidine that interacts with a GG motif that are side by side instead of spaced. These have two arginines that have another G space G. So they'll each stick to this motif. So the DNA binding motif of the zinc finger is going to recognize a G, C, G, 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 T, G, C, G motif. And wherever that motif exists in the DNA, this DNA binding domain is going to stick to the DNA there. Another common type of zinc finger domain that we see is called the homeodomain. And homeodomains are far simpler. These are much more evolutionarily old. They have what's called a helix turn helix motif, which means we have an alpha helix right here, right? Alpha helix, a turn, and then another alpha helix. So it's a helix turn helix motif, and it's this alpha helix right here that is going to get into the major groove of the DNA. Again, here's my major groove, and this alpha helix is going to slide in there and interact with the bases. We often will find that homeodomains interact with this TAAT motif of the DNA. And we find these are important in a number of places, but they're very well represented during embryological development. So again, if you take an embryology class with me, we'll discuss a whole bunch of homeodomain proteins. These are real big drivers of early development, but they're also involved in adult organisms as well. A basic zipper protein, they used to be called the leucine zipper proteins. You'll still see them called that. These guys are going to come together. They usually form what's called a dimer. So a dimer is two proteins that form a complex with each other. So this is going to be two different proteins. And they're usually called a basic zipper or a leucine zipper. The leucine zipper part comes from here. We have one protein in this dimer. It has this region up here that's rich in leucine molecules. And the other protein that forms the dimer also has a bunch of leucines. Leucines are hydrophobic. So they, in the cytoplasm or the nucleoplasm, want to get the hell away from the water. So they're going to form a hydrophobic interaction with each other to kind of give each other a hug, so that these leucines are kind of holding each other, keeping the water away from them. And that forms the two proteins to stick together and form a dimer. When they do that, these bottom alpha helices kind of stick out. I usually would jump around like I'm riding a horse in the classroom, but notice how they form like legs straddling a horse, and they're going to straddle the DNA right here at the major groove of DNA, with one leg going on this side of the DNA and the other leg going on the other side of the DNA. So they really are kind of, I think, riding the DNA as though the DNA was a horse. And these alpha helices then are going to interact with the bases here. Again, usually we'll see a T, A, and then an A, T. So that's the motif they're going to recognize. Very similar is the basic helix loop helix transcription factors. These work the same way, except they are typically not going to form, in some vocabulary, basic zippers usually form what's called a homodimer, which means it's two of the same protein coming together. What we see with basic helix loop helix are often called BHLH proteins, is they form what's called a heterodimer. Two entirely different proteins come together, and they have, again, the name here is they have a helix right here, see the helix, a loop, and then another helix right here, and this one has a helix, a loop, and then another helix. These two regions up here are going to interact with each other to form a dimer. And again, the basic region is going to straddle on either side of the DNA here and here in the exact same way. Let's say it's riding a horse. Here's the major groove of the DNA, and it's going to straddle that and make contact with the bases that are exposed here. We often see BHLH proteins are going to bind what's called an E-box, which is a palindromic sequence that's going to go C, A, anything, and then a palindrome on the other side where it's going to go anything, T, G. So notice that's the complement. A is complementary to T, C is complementary to G. So these read forward and backward the same thing, C, A, N, T, G. The complement is going to be C, A, N, T, G. So we'll often see a motif like that these guys bind. So just to kind of, and there's several more, but these are the major players. But you get the idea, I hope, of how these things work. So just to kind of show you the modularity that goes into this, one transcriptional activator generally does not activate a gene. You usually have a whole bunch of these things coming in together to help regulate a gene. For instance, what we're looking at, this colorful sequence of DNA here, this is the proximal promoter from the insulin gene. So right here that I have underlined, this is the +1 site. That's the transcriptional start site. And everything that I've underlined is actually going to get transcribed into the insulin mRNA. And then you can see here's the core promoter that upstream of that. So here's my TATA box right here. So that's part of the core promoter where this gene is going to start. Now, the proximal promoter exists upstream of the core promoter, and I've kind of color-coded some of these, but you can see these little teal boxes here, TAAT, TAAT, they bind to a protein called pancreatic and duodenal homeobox protein 1, or PDX1. It's a homeodomain protein. We said those homeodomains like to get their alpha helix in there and stick to those TAAT motifs. Now we have here, this green one is a protein, a transcription factor called neuronal differentiation 1, or NeuroD1. It's a basic helix loop helix protein. We said they like to bind those E boxes, right? That's that E box that recognizes CANNTG. So here we go, CANNTG. So that's going to bind your NeuroD1 protein. This long gray one over here, this is a basic zipper protein called MAF-A. Full name is VMAF, muscular aponeuritic fibrosarcoma homolog type A. Fortunately, we shorten that to MAF-A, and it's going to recognize this motif and bind the DNA there. And then finally, these long yellow ones here, they bind zinc finger proteins. So this is a protein called GLIS3, binds the zinc finger domain will bind those two yellow regions. So all together then, these 4 transcription factors sit down at this promoter and help recruit TF2D to this TATA box to initiate transcription at this point. So these usually work together as a group. You usually don't see one transcription factor responsible for driving this. So if you're going to be a transcription factor, then you've got to have a DNA binding domain. And then the other domain that you've got to have if you want this job is once you bound the DNA, you've got to be able to turn the gene on or off. You have to either activate or repress transcription. So activation or repression of the gene can come in a couple different ways, which if you haven't guessed, we're going to talk about in some detail. But it can either directly interact with the general transcriptional machinery, things like TF2D, TF2E, etc., or it can work indirectly with things that are called co-activators and co-repressors, which are other proteins that interact with the general transcriptional machinery. So a transactivation domain is what we call an activation domain. It's usually called a TAD, a transactivation domain. And they often, although not always, they have a lot of acidic bases in them, things like glutamic acid and aspartic acid. They're very rich in glutamines and prolines, but again, not always. So they're not the easiest thing to spot, but you can kind of scan a protein for these types of qualities, and it's often going to be indicative of a transactivation domain. Repression domains, that's completely up in there. They usually have to be identified by functional analysis because there's no way to really recognize just by looking at the amino acids what a repression domain is. So to kind of give you an idea of how this looks, that protein I was talking about, GLIS3 there, has a zinc finger DNA binding domain located kind of centrally, and that's what binds to a consensus sequence in the DNA to allow it to bind to the DNA. And then it has a transactivation domain over here at the C terminus that is going to allow it to activate certain genes like insulin. A couple of other members of this family, you notice GLIS1 looks almost exactly the same as GLIS3 with a zinc finger domain and an activation domain. It activates a different suite of genes in different tissues. And then GLIS2, notice, also has that centrally located DNA binding domain, but it has a repression domain over at the C terminus, another repression domain at the amino terminus, and a weak transactivation domain also at the N terminus. So these things can be, pieced together in any way. The DNA binding domain doesn't have to be in the middle. The activation domains can be anywhere, but you're going to find these domains are going to comprise these types of proteins. And again, to be a domain, if I take my GLIS1 protein and cut out this zinc finger domain from the rest of the protein and put it into a cell, it's going to bind DNA. So the domain can work independently of the rest of the protein. So once these transcription factors bind using their DNA binding domain, there's basically two ways in which they can activate or repress a gene. The first way is, you can see here, again, as we showed the insulin promoter, these factors don't typically bind by themselves, but form complexes of different transcription factors that all get recruited to the combinations of enhancers and the proximal promoter and the distal promoter. And this forms a large multi-protein complex called the enhanceosome. And the enhanceosome includes not only transcriptional activators, in this case binding to enhancers, and sometimes these enhancers can be hundreds of thousands of base pairs away from the core promoter. But these activators will bind using their DNA binding domain, and then they can either directly interact with the general transcriptional machinery. So we see here's TATA binding protein, or TF2D, TF2B, TF2F, E, and H. They can directly or indirectly interact with these through protein-protein interactions. So shown here, we have some co-activators. These are proteins that don't bind the DNA, but form a bridge between your activators that are bound to the DNA and the general transcriptional machinery that's bound to the DNA. So these proteins will all form one big complex that helps recruit the TATA binding protein and the general transcriptional machinery, or help recruit RNA polymerase to the promoter to initiate transcription. And in the absence of these activators, it's very often the case that the general transcriptional machinery will not be able to stay on the core promoter long enough to activate transcription. It kind of binds and falls off quite quickly. So it can only be stabilized to initiate transcription when these activators are in place. And then likewise, if you're a repressor, here a repressor can come in and bind. Often you'll see they change the term of an enhancer to a silencer if a repressor binds it. It's the same concept. It's a DNA domain that binds to a transcription factor that represses activity. And what these repressors can do is they physically inhibit the recruitment of the general transcriptional machinery to the core promoter. So if you have a repressor bound to this site, it actually blocks these proteins from binding. It kind of keeps them away and blocks their binding to repress transcription. And just to stress, these don't have to bind in the immediate vicinity of the core promoter. Sometimes you'll see 200,000 to 300,000 base pairs away. You'll see an enhancer or a silencer that regulates the core promoter. And the reason for this is DNA, we always see it drawn as, a straight linear molecule, but remember DNA is an extremely flexible molecule. So it's all rolled up and tightly around in a ball. So if I have a segment of DNA with an enhancer here, it can regulate a promoter over here because this whole thing folds up into a ball, and that puts those two regions of DNA right side by side with each other. So because DNA is so flexible, it allows for sites that in a linear model seem like they're very distant can actually become side by side when you fold it up. You also have these co-activating proteins in the form of things like mediator or CBP or P300. And these are multi-subunit large proteins. I mean, these things are usually in the 200-300 kilodalton range. They're giant proteins that, if you ever have a circle of friends where, you got this circle of friends where there's, I don't know, cliche Breakfast Club stuff. You got the jock and you got the cowboy and you got the cheerleader and you've got the nerd and you've got the artist, and they all are held together by one common friend. That's kind of what mediator or P300 do. So these interact with the general transcriptional machinery. They interact with RNA polymerase II, and they interact with the transcription factors that bind that enhancers. So here you can kind of see a schematic. This giant purple protein here is the mediator protein. And what it does is some activator can bind an enhancer, 100,000 base pairs away from the promoter, and then interact through its activation domain with mediator. And then here you have RNA polymerase with all of your general transcription factors also interacting with mediator. And then you have some additional activators down here in the proximal and distal promoter that are also interacting with mediator. So mediator is kind of the glue that holds all of these things together to allow RNA polymerase to sit down here at the core promoter and initiate transcription. So often within an enhanceosome, you'll see these large multi-subunit proteins like that. The other way that you will find that transcription factors can regulate gene activity is not through direct or indirect interactions with the general transcriptional machinery or RNA polymerase itself, but through the regulation of chromatin structure. And if you'll remember, when we were talking about DNA back in the first lecture, I had mentioned that the DNA gets wrapped around these histone proteins, and that serves several functions. One was to compact the DNA to fit into the nucleus, which is what we focused on in that lecture. But I also stressed that wrapping up the DNA around the histone proteins will allow us to protect the DNA. And I said, we'll come back to haunt you, we can use this wrapping of the DNA around histone proteins to regulate gene activity. And that is exactly what a lot of transcriptional activators and repressors do, is they modify the chromatin structure to help regulate the activity of an underlying gene. Let me show you what I mean. So here's the figure we looked at back in lecture 1, where we have our DNA wrapping around these histone octamers to form the nucleosome. So let's hypothetically say then, here I drew in let's say our TATA box is located within this stretch of DNA. So now I've got a stretch of DNA with a TATA box that is wrapped around a histone. And when it's tightly wrapped around the histone, there's really no room for my general transcription factors to come in and bind to that TATA box. So TF2D really can't interact with the DNA there because it's so tightly wound up around this histone. So in this case, this tightly wound up region of the core promoter is going to result in the gene in that vicinity be turned off, right? This gene cannot be transcribed because there's no way to get RNA polymerase II in your general transcriptional machinery into that region because of the nucleosome. So enter our transcription factors who can regulate gene activity by altering how tightly wound up the DNA is to the histone. So to give a little more in-depth picture of these histones, I said they look like little balls when you form the octamer, and that's true. But what I didn't mention is they have these little tails that hang off of them. So on the amino terminus of each of the histones, you have this little stretch of protein that kind of comes out here and forms a tail that is independent of the DNA. See how it's kind of hanging out like that? And H2 and HB also have a C-terminal tails in addition to their N-terminal tail. But these tails are made out of protein, right? And protein is subject to post-translational modifications. So we can come in and modify the tails of these histone proteins, and in doing so, we change the chemistry of the protein. So remember when we said the chemical modifications that you can do to a protein post-translationally can either activate or inactivate or somehow change the state of the protein? Well, that's exactly what we're doing here. If we generally add what's called an acetyl group, remember we said one of the chemical modifications you can do post-translation is acetylation. If you acetylate the tails of these histones, you can relax the DNA and make it less tightly wound around the histone protein. And generally speaking, if you methylate those tails, another one of our post-translational modifications, you can condense the DNA and wrap it up even more tightly around these nucleosomes. So let's start looking at histone acetylation. Again, remember that acetylation is a common chemical post-translational modification you can make to a protein. Acetylation almost always happens at a lysine residue. So we are going to add an acetyl group to a lysine residue within the tails of these histone proteins. And what that's generally going to do, remember DNA has a negative charge around it. So we have our negatively charged DNA. Let me go to our whiteboard here so I can show you what we're doing here. So we have our histone proteins like so, and they have these tails that are sticking off of them. And then our DNA comes in, and we're going to take our DNA, wrap our DNA around this histone like so. Now the DNA has a negative charge thanks to that sugar phosphate backbone. So we have a negative charge consistently around our DNA. Remember, DNA is deoxyribonucleic acid. Acids have a negative charge. Now within those tails, we've got lysine residues. And lysines are positively charged amino acids. So if we have lysines within those tails, that negative charge on the lysine is going to be drawn magnetically to, the positive charge on the lysine is going to be magnetically drawn to the negative charge on the DNA, and it's going to cause that DNA to wrap really tightly around the histone because of the opposite charges. So what we can do then is we have a protein come in that can acetylate the lysines within the tails of the histones. So we're going to come in here to this lysine and add an acetyl group. And the acetyl group is going to neutralize that charge so that it's less positive. So we can add another one here, we can add another one here, we can add another one here, and that's going to neutralize those positive charges such that the DNA is going to be less attracted to the histone because it's now less positive. So what we're going to do then, when we acetylate our histones, is if we have a region of tightly wound DNA here, let's say we have a Tata box located right here that is inaccessible. If we acetylate the tails, the lysine residues on the tails of these histones, it unwraps the DNA. So now our Tata box has kind of shifted over to here, where RNA polymerase can now get in and transcribe the gene. So this is going to result in activation of the gene. So we generally think of histone acetylation as an activating modification. In particular, lysine number 9 of histone 3, which is abbreviated H3 for histone 3,
K is the symbol for lysine, so H3K9 acetylation, and similarly H3K14 acetylation are really well known to be modifications that result in the activation of a gene. If you want to see, this is kind of a map of looking at your histone tails. So these are each of the tails that are sticking off of these histone molecules. So if we go up to histone 3 lysine 9 is often acetylated when you want to activate a gene. And likewise, so is histone 3 lysine number 14. So we often will add acetyl marks to those lysines, and that will result in the activation of a gene, generally speaking. Now, on the flip side, adding a methyl group to the lysine residues in the histone tail generally are going to have a negative effect on gene activation. Now, methylation is not quite as straightforward. So with acetylation, we're simply neutralizing that positive charge in the lysine. Methylation works differently. The methyl group can generally do a couple of two things. The methyl group can block the acetyl group from being laid down. So now we cannot set an acetyl group on the lysine because there's a methyl group in the way. Or that methyl group can kind of act as a beacon to bring in other proteins that often will block any further modification to the nucleosome and prevent the relaxation of it. But in some cases, you can actually have methylation that results in activation of a gene. So methylation is a little more gray, but generally is associated with repression of a gene. The other thing that's different from acetylation is methylation can be done in tandem. So we can have monomethylation where we add a single methyl group to a lysine, dimethylation where we'll add two methyl groups to a lysine, or trimethylation where we'll add three methyl groups to the same lysine. And oftentimes we'll see a case where like dimethylation is associated with an active promoter and trimethylation is associated with an inactive promoter. So methylation is definitely a little more complicated, but the characteristic marks for methylation is methylating lysine 9 of histone 3 with a trimethyl mark. So that's called H3 for histone 3, K9 for lysine 9. Methyltransferase will add a trimethyl group to that lysine, and that often will shut down a gene. It turns the gene off. H3K27 trimethylation, so you have histone 3, lysine number 27 trimethylation, is really going to see in particularly during development. And if you have a particular cell type that's never going to express a gene, again, go back to our example of insulin within a neuron, right? A neuron is never going to make insulin. You will often see the regions around that gene then will be H3K27 trimethylated because it's permanently turned off in that cell. And then, like I said, sometimes you see methylation as a mark for activity, H3K4, so histone 3, lysine number 4 trimethylation is often associated with an active promoter. So again, we're looking mainly at histone 3, but histone 3K9 can be trimethylated to silence the gene. K27 can be trimethylated to silence a gene. And then K4 here can often be trimethylated to activate a gene. So this is activating, and these two are both repressing. So if you look then at what we call the epigenetic landscape around different promoters, you can often spot the promoter simply by looking at the modifications that are made to these histones. So here we see a gene in which there's a promoter here, and this gene is active. So this gene is currently being transcribed. And if you look then, you see that you have histone 3 K9 acetylation is extremely high, as is histone 3K4 trimethylation. He said both of those are activating marks, so those are really high levels of that, suggesting that this gene is currently being turned on. Now, if you look at a different region of the genome over here, this gene is turned off. This gene is silenced. So here you see large levels of histone 3K27 trimethylation and some levels of histone 3K9 dimethylation. This suggests that the gene is currently off. So there's no transcription happening in that region. So tying this all together then, activators, transcription factors that act as activators, in addition to have a DNA binding domain, often have what's called histone acetyltransferase activity, or what we call HAT activity, histone acetyltransferase, HAT activity. And what happens is these guys will bind to the DNA using their DNA binding domain, and then they'll use their HAT activity to relax the chromatin in that region to allow the general transcriptional machinery and RNA polymerase II to come in there and activate the gene. Transcription factor 2D, TATA binding protein, actually will interact with an acetylated histone that then further comes in and acetylates more histones to keep the promoter open. Kind of imagine it's coming in there and holding it open while RNA polymerase is going in there and activating the gene. And then, as you can imagine, repressors will often come in and bind to the DNA using their DNA binding domain, and they have what's called histone deacetylase activity, or HDAC activity. And what they're going to do then is they come in and bind to the DNA, and using their histone deacetylase activity, they remove histones from the nucleosomes. And if you remove a histone, it's going to cause the DNA to wrap up more tightly, shutting the gene off. So this is a kind of reversible thing to fine-tune gene activity. So I have three panels I drew here. One, we have kind of a baseline level of gene activity. So there's, small levels of gene activity happening in this case. The promoter region is, moderately wrapped up around the nucleosomes. You got a couple of acetyl marks that are on these histones. And then if I have a transcription factor come in and bind to its enhancer, and it has, or in this case, a silencer, and it has HDAC activity, it's going to strip those acetyl groups off of the lysine residues on the histone, and that causes the DNA to wrap up even tighter around these histones, and that's going to effectively shut the gene off. So transcription is going to stop. But if I want to turn the gene on, I can have a activator bind to its enhancer site that has HAT activity. And the HAT, the histone acetyltransferase, is going to add even more acetyl groups to these lysines on the histones, opening up or relaxing the nucleosome. And this is going to cause major amounts of gene activity, or turning the gene full on. So we can, kind of modulate in between these states where we have, either no gene activity on up to full gene activity by, regulating how many acetyl groups and methyl groups are located on my histones. In addition to transcription factors doing this directly, we also have what's called chromatin remodeling complexes. And these are protein complexes that their job, they're not just inadvertently adding histone acetyltransferase or histone deacetylases. What they're doing is, it's kind of the road crew, they're construction crews, and they're going out there and remodeling the nucleosome, usually in an energy-intensive way. So there's a couple of different classes of chromatin modeling complexes. The first one, one of the most popular, common ones, is the SWE-SNF complex. And the SWE-SNF complex, what it does is this, I think of kind of like the bulldozer. It comes in there and binds to the DNA around a promoter that's wrapped up around the nucleosome. And it's going to actively burn ATP. And as it burns ATP, it's going to kind of bulldoze forward, and literally it's kind of the strong man on the team here. So if this is a superhero team, ZeRuth is your strong man, and it pushes the nucleosomes aside. It just pushes them open like that. And while it's holding these nucleosomes, see, it's kind of pushing these nucleosomes on either side here. And while it's holding them open, RNA polymerase can dip down in there, bind with the general transcriptional machinery, and start transcribing the gene. So these are oftentimes recruited by the transactivation domain of some other transcription factor. So a transcription factor can bind at its enhancer, and its transactivation domain will recruit ZeRuth to help, slide those nucleosomes aside and allow this gene to be activated. SWR1 complexes are another form of chromatin remodeling complexes. And so if ZeRuth is the strong man, these are kind of the brains behind the operation. What these guys do is they're the tricksters. This is the Loki of the complexes here. Normally, remember, your histone octamers are made up of H2A, H2B, H3, and H4. Well, SWR1 comes in and stealthily replaces H2A with a very similar look-alike protein called H2AZ. And H2AZ is not as stable as H2A. So it's like taking the steel bead that's H2A and replacing it with a glass one. So that's going to create a very unstable nucleosome that as soon as RNA polymerase or anything else comes in there and touches it, it just shatters and falls apart. And you can allow for the gene to be turned on. So we'll often see SWR1 work on genes that we call are constitutively expressed, which means they're always on. So if you think of things like tubulin or actin, some of the genes that make the proteins that make up our cytoskeleton, these genes are always on. You're constantly making them. So they're often going to have H2AZ replacing H2A in those nucleosomes to make sure they can never get shut off. And then finally, ISWE, if ZeRuth is the bulldozer, ISWE is kind of the steamroller. It comes in here, and this is going to work in the opposite direction. So instead of activating a gene, ISWE will work to repress a gene. And what this does is it basically just rolls through a region of DNA and just places nucleosomes one after another. So it's going to nucleosome here, nucleosome here, nucleosome here, nucleosome here, just completely covering the area with nucleosomes so that no gene in that region can be turned on. And this will silence a gene within that region. So while SWR1 is going to be kind of characterized by having constitutively active genes, this is the opposite. This is regions of genes we want to silence, where we're not going to turn these genes on. They're just hopelessly silenced in that cell. So let me segue here for a second and get back to our original conundrum then. So our original conundrum was, if transcription is initiated by the promoter and all cells within an organism have the same DNA, then all cells have the same promoters in front of each of their genes, and they're going to bind to the general transcriptional machinery in all cells that there are. So there's no way you could differentially transcribe a gene, because all of the cells have the exact same promoters in front of the genes and therefore going to activate identically. So we said, okay, well, we're going to solve that problem by putting a proximal distal promoter and enhancers in the DNA. But we have the same issue here, right? Those proximal distal promoters and enhancers are still made up of DNA. And if every cell has the same DNA, then every cell is going to have the same proximal distal promoters and the same enhancers, and they're going to bind to the same transcription factors and be regulated identically. So we really haven't solved our issue of how insulin can be expressed in a beta cell but not in a neuron, right? They have the same DNA, they have the same sequences. There's a story that I'm sure is more urban legend than anything. But an astrophysicist was giving a lecture on the universe, and he was discussing, the effects of gravity on holding solar systems together and that type of thing. And the legend goes that a woman in the audience said, "That's nonsense. These are not the forces that hold the planets, Earth in place. Earth is resting on the back of a giant turtle." This is actually a religious belief that some people will have, that the Earth sets on the back of a giant turtle. So the physicist, thinking he's got the woman, said, "Well, my dear lady, if that's true, if the Earth is resting on the back of a giant turtle, then what is the turtle resting on?" And she said, "Well, don't be ridiculous. There's turtles all the way down." And that's kind of where we are right now, right? So the core promoter is made of DNA, therefore cannot be regulated differently in each of the cells if each of the cells have the same DNA. But the core promoter, we said, is regulated by the proximal distal promoter, which is also made of DNA. So we're also going to have the same proximal distal promoter in all of our cells, and therefore have no way to differentially regulate a gene. So this time I promise I get to the end of this infinite turtles all the way down loop. The transcription factors, the proteins that bind to the enhancers in the core and proximal promoters, they too are regulated. So we can ultimately regulate whether one of these transcription factors is on or off. And that is going to dictate whether a gene is on or off within a particular cell. So let me give you a couple of examples of how this works so that you can kind of put all this together. This is by no means exhaustive. There are tens of thousands of different genes that are involved, but these are a couple of characteristic ways this is done. And you're going to see in this class and all of your subsequent classes you take beyond this class that we're going to get variations on a theme, right? Once you kind of get a general method for how something done, everything else is some variation of that theme. So let me give you the first major variation on a theme, and this is what's called the nuclear receptors. The nuclear receptors is a group of transcription factors, and it's largely comprised of hormone receptors. So this includes your estrogen receptor, progesterone receptor, androgen receptor, cortisone, things like that. All of these hormones are going to be, these are receptors for hormones that signal different events to the cells. And the way the nuclear receptors are regulated, here we're going to look at estrogen receptor as an example, but they all work the exact same way. So here, estrogen receptor, this is our transcription factor that has a DNA binding domain, and it has a transactivation domain. So it's a classic transcription factor, but it exists in the cytoplasm bound to two chaperone proteins called heat shock protein 90. If you remember, heat shock proteins are molecular chaperones. We discussed HSP70 in the last lecture that help proteins fold properly. But in this case, these two molecules of heat shock protein 90 are bound to the estrogen receptor, and they are blocking a nuclear localization sequence. So behind that heat shock protein here is an NLS that is inaccessible right now because heat shock protein's in the way. Now, nuclear receptors, remember I said the bare minimum you need to be a transcription factor is you need a DNA binding domain, and you need either an activation or a repression domain. But some transcription factors have additional domains that go above and beyond. The nuclear receptors have a third domain called the ligand binding domain. And the ligand binding domain, as the name suggests, is a domain within the protein that binds to some ligand. A ligand is a molecule or a protein that activates something. So my estrogen receptor is sitting in the cytoplasm, bound to these two molecules of HSP90, and they are blocking the ability of estrogen receptor to go into the nucleus. So in this case, estrogen responsive genes are turned off because estrogen receptor, the transcription factor that turns them on, is not in the nucleus where the DNA is. So if it's not in the nucleus, it cannot activate estrogen responsive genes. You with me? All right, so the ligand that's going to activate the situation then is going to be, if you haven't guessed, estrogen. So if estrogen enters into the cell, estrogen binds to the ligand binding domain, and that causes the protein to kind of change its shape a little bit, and it ejects HSP90. So HSP90 now leaves. When HSP90 leaves, that exposes the nuclear localization sequence. So now with the NLS exposed, remember we're going to have important. Here's our important protein. It's going to come in here, and important is going to bring this into the nucleus, where now once in the nucleus, it can bind using its DNA binding domain. It can bind to the DNA, and using its strand activation domain, it can activate target genes and turn estrogen responsive genes on. So even though the promoter region, the enhancer that binds to estrogen, is present in the DNA of all cells, the genes will only ever be turned on in cells that are exposed to estrogen to activate the system. So that is one way of doing it. That's a common way of doing it, is to sequester the factor in the cytoplasm. Therefore, it's not active unless some ligand or some signal that tells it to be active is present. So estrogen will activate estrogen receptor, testosterone will activate androgen receptor, progesterone will activate progesterone receptor, and you get the idea. We can also, remember, transcription factors are proteins. So we can post-translationally modify our proteins through any of the ways we've talked about, right? Phosphorylation, acetylation, methylation, ubiquitination, sumoylation, glycosylation, proteolytic cleavage. Any of these things can activate the transcription factor or turn it off to regulate gene activity. So I'll give you an example of, again, this is extremely common, but variations on a theme. One example of how this works is what's called JAK-STAT signaling. JAK-STAT signaling is a really important signaling mechanism. It's involved in development. It's involved in cancer. It's a widely used signaling mechanism. And what happens here is you have a transmembrane protein. Remember, these are proteins that are synthesized at the ER that move over to the plasma membrane. So we have these two transmembrane proteins that act as receptors. And these are receptor tyrosine kinases, which means they're receptors that get phosphorylated on a tyrosine. And they are going to be kind of at some distance from each other in the plasma membrane when they're off. And then a ligand, in this case an interferon, is going to come in and bind to the receptors here. And when the ligand binds, it takes the two receptors that are in the membrane and it draws them together like this. So now they get drawn in towards each other when the receptor is bound to its ligand. And what that's going to do is on the cytoplasmic face of the receptor, you have these proteins called JAKs. And when they get drawn in next to each other, this JAK is going to phosphorylate this JAK. And this JAK is going to phosphorylate this JAK. So they kind of give each other a big smooch and phosphorylate each other. And then once they become phosphorylated, then this JAK will phosphorylate the receptor in the cytoplasmic tail, and this JAK will phosphorylate the receptor on its cytoplasmic tail. And that phosphorylation works like a homing beacon, and it calls in another protein called STAT. And the STAT protein is going to bind to that phosphoryl group on either side of the receptor, and then the JAKs that were up there are going to phosphorylate the STAT once it's bound. And once you get a phosphorylated STAT protein, they're going to kind of jump off of the receptor and go hug each other and form a dimer. And the phosphorylated JAK dimer now moves into the nucleus where it can bind DNA using its DNA binding domain and activate genes using its transactivation domain. So again, this system, these genes that are regulated by JAKs and STATs are going to be turned off so long as STAT is not phosphorylated, and STAT only gets phosphorylated when some signal from outside the cell, in this case an interferon, activates the whole system. So those are just two general examples of how this works. Again, all variations on a theme, but you're generally going to have the transcription factor activated by some signal that binds to some receptor within the cell, and upon activation, it can then bind to the enhancer where it activates or represses the gene. Genes can also be regulated through chemical modifications of the nucleosome, which we discussed, or of the DNA itself. So independent of the histone, DNA itself can be directly methylated by proteins called methyltransferases. And we generally associate high levels of DNA methylation, what we call hypermethylation, with very low transcriptional activity. The idea is here, if I have a stretch of DNA and I put methyl groups all over it, the transcription factors can't bind anymore, right? It just blocks the area. Low levels of methylation, we call hypomethylation, therefore, is associated with high transcriptional activity because transcription factors can now access the DNA and bind to it. So these chemical modifications of both the DNA and the histones collectively are called the epigenome. And epigenetic studies have really become a hotbed of biomedical research. And in your somatic cells, your non-sperm or egg, all of your normal body cells, these epigenetic changes to the DNA and the histones are preserved as the cell divides. So as the cell duplicates itself into two daughter cells, the same epigenetic changes are inherited from one generation of cell to the next generation of cell. In your sperm or your egg, on the other hand, your germ cells, they undergo a clean slate in a process called reprogramming, where all of the epigenetic modifications that have been acquired throughout the cell's life are wiped clean and erased. So a recent area of research has been looking into this, and they found in some cases, for reasons that aren't entirely clear yet, these epigenetic marks are not always wiped off of the sperm and the egg. So therefore, there are some levels of what they're calling epigenetic inheritance, where you can inherit a change to the DNA from your parent, or in some cases, your grandparent, which, if you remember, we were talking about Jean-Baptiste Lamarck, who said traits were acquired by changes that happened to the organism during its lifetime. If this is true, and it looks to be that there's some truth to this, he was in part right. So we get, yay, you get vindicated. You were actually kind of right in that. In some cases, we do see what's called epigenetic inheritance. And the consequences of this are being investigated, but some of the early studies seem to have some pretty profound consequences for inheritance, in which, let's say, you have a mother that smokes cigarettes, not even while she's pregnant. Let's say she smoked 20 years before she got pregnant. Smoking cigarettes leads to high levels of epigenetic changes to the DNA. So if you smoke, you're going to be adding methyl groups to your DNA that are not generally found in non-smokers. Well, when you fail to completely reprogram the egg cell, some of those methyl marks will get passed on to the next generation, your child. And it turns out it looks like those epigenetic marks that did not get erased from your cigarette smoking can lead to disease states in the child. So they've associated some of these epigenetic marks that get passed on with deleterious effects in the children. In fact, the one study showed that this was passed on not from the mom, but from the grandmother. So the maternal grandmother passes on her epigenetic marks that were acquired through smoking to the grandchild. Um, and they're similarly finding these types of things with obesity. Obesity is another thing. If you're overweight or obese, it causes a number of epigenetic marks to get added to your DNA due to the stress. And those too appear to be inherited. In fact, there's some reports suggesting that epigenetic inheritance from obesity in the mother might have some contribution to autism, although that's still yet to be investigated. So this means that environmental interactions with the parent could affect the genetics of the offspring. But again, this is a kind of relatively new area of research. There's a lot of unknowns here. A lot of it may be overblown, but within, we'll figure it out eventually as to what consequences has. But that being aside, genomic imprinting occurs when a parent-specific allele is permanently turned off through an epigenetic modification, usually a methylation. And this is extremely rare, right? There's only a couple of genes where this is an issue, but just to show you how this works, the genes for insulin-like growth factor 2 and H19 are both located on the same chromosome. And in the maternal chromosome, the chromosome that you inherited from your mother, the insulin-like growth factor 2 gene is permanently turned off due to what's called an insulator protein that binds to an insulator region. And an insulator protein is a protein that binds to the DNA and basically forms a wall. Trump would like this one. And this wall is gonna sitting here, and it blocks a transcriptional activator, which binds to an enhancer. Here, it blocks its ability to activate this gene because the wall's in the way. So in this case, in the maternal chromosome, the transcription factor binds to the enhancer, and it can activate transcription of the H19 gene, but it cannot activate transcription of the insulin-like growth factor 2 gene because the insulator is in the way. So on mom's chromosome, H19 is on, but IGF2 is off. Now on your paternal chromosome, the one you got from your dad, we have epigenetic modifications of the DNA in the region of the insulator region and the H19 promoter, which we add a whole bunch of methyl marks to the DNA. So we methylate the DNA. And like I said, when you methylate the DNA, it prevents other things from interacting. So in this case, my insulator cannot bind because methyl marks are in the way. And also, my activator protein that binds here cannot recruit RNA polymerase to the H19 promoter because the methyl mark is in the way. So in the paternal chromosome, H19 is off because the promoter is methylated. But since the insulator can't bind here, this now can activate IGF2, which is on. So you have IGF2 on dad's chromosome and H19 off, and you have H19 on in mom's chromosome, but IGF2 off. And then when this individual goes on to produce their sperm or their egg, they're going to reprogram these. And they're going to completely wipe clean all of those epigenetic marks. So that means in the germ line, in the sperm or the egg, the the insulin-like growth factor 2 and the H19 are both on, right? On both of your chromosomes. So on mom's chromosome and dad's chromosome, they're on because you reprogrammed them. And then as you become a male or a female during development, if you have a Y chromosome or an X chromosome, the sperm are going to go ahead and get epigenetically modified again. So in the sperm, you're going to have methyl marks along here, keeping H19 off, but IGF2 on. And in the egg, you're going to wind up having an insulator here, which is going to allow H19 to be on and IGF2 to be off. And then when the sperm and the egg fertilize, you get right back to where we started, where your paternal chromosome has IGF2 on and your maternal chromosome has H19 on. So that just happens generation after generation after generation, keeping this reprogramming going. All right, finally, wrapping this up then, as we've kind of been doing traditionally, we're going to look at how prokaryotes do this, your bacteria. And again, as always, prokaryotes do it slightly simpler. Simpler, is that a word? I guess it is simpler. We talked about all of these different, we have leucine zippers or basic zipper proteins, basic helix loop helix proteins, zinc finger proteins, all that are different types of DNA binding domains. Most bacteria transcription factors have a helix turn helix DNA binding domain. So they generally use the same type of DNA binding domain across the board. This looks, again, you have a helix here, so there's your helix, a turn, and then another helix, and here you have a helix, a turn, and another helix, and it's going to do that horse riding thing again, where this helix is going to straddle the DNA on this side, this one will straddle the DNA on this side, and these are going to bind here in the major grooves of the DNA, and they generally recognize what we call an inverted repeat. So notice this one goes G, A, A, T, T, G, T. Then the other side, it goes G, A, A, T, T, G, T. So it's this inverted repeat in two directions. That's what these helices are going to stick to and bind to the DNA. Another major difference between eukaryotes and prokaryotes is prokaryotes have what's called an operon. And an operon is a cluster of related genes that are all regulated by a common promoter. So it's one promoter that makes one mRNA, but the one mRNA they make has multiple open reading frames for different proteins. And these proteins are invariably always involved in the same metabolic pathway. So when you have a single mRNA with multiple open reading frames or recipes for different proteins, we call that a polycistronic mRNA. And as far as we know, eukaryotes do not do this. This has only been identified in bacteria. And the promoter region that regulates the operon is made up of two main parts: the promoter, which is where RNA polymerase binds, and the operator, which is where the repressor binds. So let's take a look at this. This is a polycistronic mRNA. So in this case, the Shine-Dalgarno is the bacterial equivalent of the Kozak sequence. So this is my Kozak sequence start codon. So this is one mRNA. This is my 5' end, this is my 3' end. I have a start codon here and a stop codon here. So that makes one protein. And within that same mRNA, I have another start codon here, another stop codon here. That makes a second protein, a different protein. And then I have a third start codon here, third stop codon here. That makes a third protein from one mRNA. So a polycistronic mRNA is one RNA transcript regulated from one promoter, that in this case is going to make 3 different proteins through 3 different open reading frames. So let's go ahead and look at an example of this. The classic example of an operon is what we call the lac operon, and this is used in E. coli. E. coli, one of the characteristic things about the E. coli species is that it can digest lactose instead of glucose, or in addition to glucose. And the reason it can eat lactose or break down lactose is because of the genes that are expressed through the lac operon. The lac operon produces genes that are responsible for lactose metabolism. And one of the main genes involved here is this gene here for beta-galactosidase or beta-galactoside. And what that does is, this is lactose, it's a disaccharide. This gene comes in here and cleaves these 2 rings in half, yielding 1 molecule of galactose, which can then be converted into glucose, and another molecule of glucose that can be used. So it's extra work. E. coli would rather use glucose if it has it, but if glucose isn't available, it can break down lactose if it needs to. So the lac operon then is going to have a promoter region, which is going to bind to RNA polymerase to initiate transcription to make a polycistronic mRNA that produces the genes, the open reading frames, for 3 proteins: LAC-Z, which makes beta-galactosidase, LAC-Y, which makes an enzyme called permease, and LAC-A, which makes an enzyme called transacetylase. So all 3 of those proteins are encoded for by the same mRNA on this polycistronic mRNA. So you have a promoter region here that binds to RNA polymerase, and in front of that you have this is an operator region which can bind to a repressor. And the way this generally works for an operon is, you can kind of think of it pretty simply like this. If I have my operon, and this is my transcriptional start site, here is my promoter, and that is going to bind to RNA polymerase to initiate transcription of this gene. The operator kind of sits in front of the promoter, and that binds to a repressor protein that basically gets in the way. So if the repressor is bound to the operator, it doesn't allow RNA polymerase to move forward to the gene and transcribe it. So if the repressor is bound to the operator, the gene is usually off. This is Gondorf, you shall not pass, Balrog. So that's the general gist of what we're going to see. It's quite simple in bacteria. A promoter simply binds RNA polymerase. The operator simply binds the repressor. And yeah, I made a comment about a Balrog of Morgoth because I'm a nerd. Let's see. So let's go ahead and look at how this works then. If glucose is present and lactose is not present, then there's absolutely no reason for the E. coli to produce these genes, your LAC-Z and your LAC-Y and your LAC-A. Why waste the energy making these enzymes to break down lactose if you don't have any lactose? So in the case where glucose is present and lactose is not present, then you're going to have a repressor protein that comes up and binds to the operator and blocks RNA polymerase from transcribing, and the gene is off. Okay, easy enough, right? Now, for the on state, we have to add an additional level of regulation for this, because like I said, if the E. coli has glucose present, it would rather eat the glucose and not even mess with the lactose. So it really only wants to turn it on if you have lactose present and no glucose. Because if glucose is present, it's not going to waste its time with this. It'll just eat the glucose. And the way it can sense its environment, that's the whole idea of gene regulation, is you're sensing your environment. It knows how much glucose is present, if I can anthropomorphize it, simply through the chemistry of glucose metabolism. Remember from Bio 115, if you have high levels of sugar, glucose, in the cell, then it's going to convert adenylyl cyclase, or it's going to convert cyclic AMP into ATP. So in this case, you're going to have high levels of glucose, mean you have low levels of cyclic AMP, and that's going to indicate to the cell that you have high levels of sugar. If, however, you have low levels of glucose in the environment, then that means you're going to have high levels of cyclic AMP present because it's not being turned into ATP, and that is going to wind up telling the cell that you have low levels of sugar and therefore low levels of glucose and therefore need to metabolize lactose. So that's going to be our final switch we add to the lac operon. So if you have no lactose present, the repressor is going to bind to the operator and shut the whole thing off. If, however, you do have lactose present, but glucose is also present, we have two things that happen. One is, since we now have lactose present, lactose is going to bind to the repressor and prevent it from binding the operator. So now our operator is wide open, which means RNA polymerase can bind here and, in theory, move forward because it's not being impeded by the repressor. However, since we have very low levels of cyclic AMP, because we have high levels of glucose, cyclic AMP cannot bind to our third element called the cap-camp binding site. And this cap-camp protein would want to bind here only if cyclic AMP is present. But in this case, since we don't have cyclic AMP, it's not going to bind, and that's going to prevent RNA polymerase from binding and shut the whole thing off. Now, the only time we're really ever going to turn this on then is if we have lactose present and glucose is absent. So in this case, we're going to wind up with lactose binding to the operator to prevent it from binding to the repressor, to prevent it from binding to the operator. That allows RNA polymerase to sit down at the promoter. And we now, since glucose levels are low, we have high levels of cyclic AMP. And cyclic AMP can then bind to the cap-camp protein, which then binds to the cap-camp element and allows RNA polymerase to bind here and turns on the gene. So there's a three-way switch that we can do with a lac operon that all respond to their environment. If lactose is absent, then the repressor is bound to the operator and the whole thing is shut off. If lactose is present and glucose is absent, then the lactose will turn off the repressor so it cannot bind to the operator. Cyclic AMP will activate the cap-camp protein, which then allows RNA polymerase to bind the promoter and turns it on. If, however, you have lactose present and glucose is present, then the lactose will turn off the repressor so it can't bind to the operator. But since you lack cyclic AMP, the cap protein cannot get activated, and therefore RNA polymerase still can't activate the gene. So it's a three-way switch depending on what the environmental situation is. And that is about as complicated as a operon can get, which is why I give that as an example. Every other form of an operon is usually a two-way switch where you just have an operator and RNA polymerase binding to the promoter, and that's the gist of it. So that is, I know, a ton of information. So review that. And we only have one more lecture left of this unit. So I will promptly try and get our next installment on mutation up for you. And we are already heading into exam time. So I have, I will get your exam posted on Canvas with the official date shortly, but make sure you're keeping up with this. I know it's a ton of material. You need to be putting, several hours a week into studying to make sure you're keeping up with this material. And once again, if you have any questions or anything is not clear to you, get in touch with me and I'll do my best to work it through with you. And I will see you on our next installment.